Hello friends, I am Suchi. Welcome to my YouTube channel. In this video, we will discuss about antibody. In previous videos, we have discussed about antigen, any substance which is not belongs to a particular tissue or organism. When that substance enter into the tissue, it will induce immune response against that particular substance, and body will produce certain chemicals against that particular foreign substance, which we are calling as antigen. Those chemicals are known as antibody. Which is specific to that particular antigen. These antibodies are produced in body serum. They are protein in nature. Normally they are globular protein. When body produce chemicals due to immune response, but they are non-specific to this particular antigen, that time those chemicals we are calling as immunoglobulin. Those are protein in nature and they are also produced due to immune response, but they are non-specific. It means they will not bind with that particular antigen, but this antibody will bind with that particular antigen. Both this immunoglobulin and antibody are same. When they will be specific, they will known as antibody. When they will be non-specific, they will not bind with that antigen, they will known as immunoglobulin. It means these immunoglobulins and antibody both are same, but all antibodies are immunoglobulin but all immunoglobulin are not antibody. Both antibody and immunoglobulin produced by plasma cell, they both produced by plasma cell which have developed from B lymphocyte. Both this antibody and immunoglobulin are serum protein. globular protein. Now we will discuss about the structure of antibody or structure of immunoglobulin. If you will go for the basic structure of antibody, it is a Y-shaped structure molecule found in body serum. It was first proposed by Rodney Porter in 1962. It contains two identical small chain which are known as light chain and two identical large chain which are known as heavy chain. On the basis of its structure and amino acid sequence, this light chain is of two types. One is K type, another one is L type. This K type also known as kappa type and this L type is known as lambda type. If any antibody contains K type of light chain, means both the light chain will be K type. If any antibody contains L type of light chain, it means both the light chain will be L type. But in body serum you will find 60% of K type of light chain containing antibody. Each light chain and heavy chain contains two terminal end. One terminal end is N terminal end or amino terminal end. Other terminal end is C terminal end or carboxylic terminal end. First we will make a structure of an antibody. These are the light chain and here are the heavy chain. In both light chain and heavy chain, you will find N terminal and C terminal. Here is N terminal, here is C terminal. Similar way, here is N terminal, here is C terminal. At N terminal side, you will find amino group. Similarly, C terminal side you will find carboxylic group. Each heavy chain is twice the length of light chain and molecular weight of light chain is 25,000 Dalton. Similarly, molecular weight of heavy chain is 50,000 Dalton. Each light chain bind with heavy chain by single disulfide bond. So, here you will find one disulfide bond which is present in between light chain and heavy chain. Both the heavy chain connected with each other at their central flexible region by number of disulfide bond. Central flexible region is known as hinge region. So, this is the hinge region which is central flexible region of heavy chain. In this light chain, you will find two intra-disulfide bond. Here you will find one disulfide bond at its variable region, and this variable region you will find at amino terminal end, and another one disulfide bond you will find at C terminal end or constant region of light chain. Similar way, you will find total 4 intra disulfide bond in heavy chain. One you will find near N terminal region which is variable region of heavy chain. Other 3 disulfide bond you will find at constant region of heavy chain. It means in light chain you will find one variable region and one constant region, and in heavy chain you will find one variable region and 3 constant region. This is the variable region, and here is one constant region, here is another one constant region, here is third constant region of heavy chain. First we will mark it. So, variable region you will find at the N terminal region or N terminal site. And here are the constant region. And in this part you will find constant region. It means in each light chain you will find one variable light domain and one constant light domain. Similarly, in each heavy chain contains one variable heavy domain and three constant heavy domain. Each heavy chain and light chain contains variable region, and in their variable region some part contains inconsistent amino acid sequence. Those region is known as hypervariable region. So, in variable region some part which is known as hypervariable region having inconsistent sequence known as hot spot. In this hypervariable region of antibody, antigen will get bind. At which point this antigen will get bind in this hypervariable region, those particular point is known as idiotypic region. Suppose this is the variable region. Suppose at this point the sequence is highly inconsistent. So, these are the hypervariable region or hot spot in this antibody. So, at this region antigen will get bind with it at this point, and at this point. So, in light chain you will find 3 idiotypic region, and in heavy chain you will find 4 idiotypic region where antigen will get bind. So, at which point this antigen will get bind with it, those particular region is known as idiotypic region. When this antibody treated with any proteolytic enzyme like papain, or pepsin, that time this antibody will break and form 2 separate pieces. One fragment or one piece is known as FAB, or you can say antigen binding fragment. Another one fragment is known as FC fragment or crystallizable fragment. This is all about the structure of antibody. Now we will discuss about the type of antibody. Based on the structure of heavy chain, these immunoglobulin are classified into 5 different types. Those are gamma immunoglobulin, alpha immunoglobulin, delta immunoglobulin, mu immunoglobulin, and epsilon immunoglobulin. Now we will discuss one by one. First we will discuss about this gamma immunoglobulin. It is much more stable compared to any other immunoglobulin. It appears Y-shaped like structure. Previously we have discussed about the structure of antibody, that is the Y-shaped structure. It contains 2 light chain and 2 heavy chain, and also having variable domain and constant domain. It produces greater amount during secondary immune response. When body will infected by any antigen or microbes, that time body will produce antibody against that particular microbes or antigen. When same body will infect by same type of antigen for second time, that time body will produce more amount of this gamma immunoglobulin, which will fight against that microbes and protect the body. Its molecular weight is nearly 150,000 Dalton, its sedimentation coefficient is 7s, and its half-life period is nearly 25 days. It is the only immunoglobulin which can cross placenta or cross human placenta from mother to baby, so that it can protect the newborn baby up to 6 to 9 months. It can neutralize different type of toxin. It can enhance phagocytosis process when it will bind with bacteria. Now we will discuss about the second type of immunoglobulin, that one is alpha immunoglobulin. This alpha immunoglobulin you will find in body fluid or body serum, and it can also secrete by plasma cell. When it will secrete by plasma cell, that time we can tell that immunoglobulin or that alpha immunoglobulin is secretory alpha immunoglobulin. You will find this serum immunoglobulin which is present in body fluid. In which part this serum immunoglobulin you will find? In gastrointestinal tract, in respiratory tract, and also in urogenital tract. Its molecular weight is nearly 160,000 Dalton, and its sedimentation coefficient is 7s, and its half-life period is nearly 6 to 8 days. But this secretory alpha immunoglobulin is much more larger compared to serum alpha immunoglobulin, because in serum case this alpha immunoglobulin present as monomer, but in this secretory case it present as dimer stage. This is one monomer, this is another one monomer. So in this case it is present dimer, and both the monomer joined with each other by secretory component and by J chain. So this is the J chain which helps to bind this monomer, and this is the secretory component which can seal both the monomer at this J chain part. So this is the secretory component, and this one is J chain. And its molecular weight is nearly 500,000 Dalton. It is secreted by plasma cell. It acts as mucosal paint or antiseptic paint to prevent adherence of microbes in respiratory tract, gastrointestinal tract, and urogenital tract. It present in colostrum, which is the first milk of mother for babies. It protect baby from intestinal pathogen. That's why doctor suggesting for breastfeeding. Breastfeeding baby is more safer compared to bottle feeding. Next type of immunoglobulin is delta immunoglobulin. It is a typical immunoglobulin having Y-shaped structure. Already we have discussed about Y-shaped structure of antibody. Its molecular weight is nearly 180,000 Dalton. Its sedimentation coefficient is 7s, and its half-life period is 2 to 3 days. It found on the surface of B lymphocytes, it do not show any antibody activities. Along with immunoglobulin mu, it bind with or it act as antigen acceptor. Next immunoglobulin is mu immunoglobulin, which is the largest immunoglobulin you will find in body serum. Its molecular weight is nearly 950,000 Dalton. Its sedimentation coefficient is 19s and its half-life period is 5 days. It appears circular pentameric structure bind by disulfide bond and J chain. Suppose here I can make, this is one monomer, this is another one monomer, this is the third monomer, this is the fourth monomer and this one is the fifth monomer. All the monomer bind with each other by disulfide bond and with J chain. So, here are the disulfide bond and chain present here to bind all 5 monomer to make circular pentameric structure of mu immunoglobulin. It is the earliest immunoglobulin to be synthesized by fetus starting by about 5 months of fetal life in mother's womb. As it present from the beginning of the life of a baby, it appears in primary immune response and it acts as useful indicator for recent infection. It is having pentamer, so it shows multiple antigen binding site. So, it is having high affinity towards multivalent antigen. It localizes in blood serum and protect against blood serum infection. Epsilon immunoglobulin is the last immunoglobulin which we are talking about. It is the typical immunoglobulin having Y-shaped structure, having 2 light chain and 2 heavy chain, and it does not have central hinge region. This is the structure of immunoglobulin. You will find central hinge region here. Both the heavy chain bind with each other by disulfide bond. But in this case you will not find this central hinge region. Its molecular weight is nearly 190,000 Dalton. Its sedimentation coefficient is 8s and its half-life period is nearly 2 to 3 days. This immunoglobulin is heat labile. Other than this, rest of the immunoglobulin type are heat stable. It is synthesized by very few plasma cell. Means this type of immunoglobulin you will find in very stress amount in body serum. This type of immunoglobulin level will increase in body serum when one person suffer with allergy. It has special attraction to basophil cell and mast cell. It produces histamine or vasoactive amine. It is also known as skin sensitizing antibody. Originally it was known as reagent. Now we will discuss about the function of immunoglobulin or antibody. It directly bind with antigen to protect body, and also it helps to activate complement system. This is all about the structure of antibody and its function. Hope you understand. Please like, share, comment and subscribe my channel. Thank you.